Hi everybody, my name is Salvatore La Rosa. I'm a PhD in chemistry and I am Vice President of Research and Development at CTF. I came to CTF 2 years ago. Annette called me from Italy. We were working together in the same company in Siena, and we were a co-project leader on some projects there, so we had past experience. And she said, would you like to come to New York working for CTF and in the field of NF? I didn't know anything about NF. I didn't know what NF was, nothing. But I accepted it, and now today I really enjoyed my time these 2 years. I like the community. I cannot say I like the disease, but I like the way that we are really making life difficult for this disease. We're trying to tackle it from many different ways, and I'm sure we are really close to a big success. And in the future, we are building the path to even bigger success. So I'm gonna read some of the questions that Chad and Bobby taught for me today, and I hope I can inspire you with some of the stories that has been so successful, so exciting here at CTF. So, in as brief as you can describe it, how are drugs developed? So this is not a very easy question because drugs are developed in a very complex way, and you need so many different skill sets and so many different individuals to bring a concept from an observation of a disease to a product on a shelf in the market. Think about starting from the patient who experienced something, and then you need a physician who understands, and then a clinician, a surgeon, extract the tissue, and then the biology, and chemists who develops targets, and from there you screen, you create model of that disease. So you need molecular biology modeler, screening experts, and then people who create and genetically modify animals which have that disease, where you can screen and see for efficacy. And then from there you need someone who is good at pharmacology and see if that drug really behaves in the right way in that body, in that organism. And from there you move to animals, and then to clinical trials. You interact with the FDA, you need regulators, you need a lot of different people, legally, market developers, market analyzers. I mean, everything really. And it's a process that takes about 15 to 20 years, so very complex. You start with one hypothesis, and the ratio of getting one thing from start to the end is about 100,000 molecules that you start at the beginning, one reach the end. So it's not easy. It's not easy. What did CTF research look like in the past? Well, CTF has been around for about 40 years, and up to about 3 or 2 years ago has been doing what other great foundation has been doing out there, funding good research. We commissioned a study to a Spanish researcher recently, a year ago, and he came back to us. He was analyzing all the data set that's been generated for NF, trying to understand some signature of that disease. So he was searching the literature extensively for everything that was out there and generated, and he had to look at all publication, all article, everything. And he came back to us saying, look, I found everything, but 70% of this data has been sponsored or co-sponsored by CTF. This for us has a great impact, because we can say that most of the literature, the data out, is funded by us. So that means that we know that our review process selects the best science and the most meaningful research that is needed. But from the past to the future, how do we want to really change it? If we go at the same pace, if we go at the same speed, we will never reach an answer in a decent time frame. So we said, how can we speed up this research process? So what we did with Annette is looking at the research landscape and trying to analyze what were the gaps in research, in NF research, and how other, for instance, research community have advanced their science and created treatment options. We took a lot of examples. We saw also how other foundations really trying to explain and reach the same goals. And what we've seen is that what was missing in NF is really collaboration. It's a collaborative effort. So what we did is trying to convince them to collaborate together, and we created those collaborative consortia called Synodos. We started Synodos for NF2 last year, and is really bringing a lot of success. It's about 8 different institutions, 12 PIs. So it's a large effort, like you guys are, at Cupid. And you can see that, if researchers are together, they're collaborating, they're brainstorming. 1-2 days, it really takes you to the next level, and it shows you the best idea you can have and enhance it, make it better, make it working better. And in a relatively short time, you come with the best strategy for your research, as you do here at camp, that you get together, brainstorm those key questions that you really need to resolve, and trying to agree all together towards a plan. So this is the future for us also. So if you ask me what is research in the future, I see more and more of this consortia coming up, consortia of collaboration. You need now different skill sets that understand from biology to informatics to big data analysis. This is becoming really a key. You need a geneticist, you need a chemist, you need biology, all the disciplines that need to come together. And it's important that all stakeholders are in the same room, so they understand each other, they understand the steps, and they don't make anything that is tomorrow cannot be translated to the clinicians, because the clinicians will be there and say, okay, look guys, you need to do this process in this way if you want me tomorrow to pick it up for the patients. So, and also, if you are in a group, you are more stimulated. You have a lot more control of what you're doing, because you know you're going to influence the work of someone else. CTF is doing its work as well, because we are bringing together those groups. And as you know, if you have many heads, you need a lot of coordination. You don't want to duplicate work effort. You don't want to do the same task over and over. And you want to be efficient, because your deadline is critical for the next person who's going to pick up from there to do their own task. So it's very important that you understand how things are connected. So we are helping them with this. We are helping them with project management. I'm the project management manager for Synodos NF2. The other Synodos NF1 effort will be managed by CTF staff. We have another 2 persons here at CTF, that one is already here, one is we're going to hire it very soon, that will manage those projects together with me, and we'll bring it to the next level, because we know and we have that experience that is required, like in an industry, to put everybody on the same schedule and progress those results faster. What is the story of AZD 6244? Well, this is very exciting. AZD 6244 is a MEK inhibitor. MEK is a class of enzyme who is involved in cellular growth and proliferation. It's very particular because some type of cancers, and in particular solid cancer, are affected by this pathway. So there is a tumor which is growing where this molecular mechanism is out of control. So cancer grows because there is a continuing feedback of signaling that will push this growing of cells. This molecule stops that process and keep it under control. This was first tested in the very first collaborative effort that we had at CTF. It's called the NFTC, the Neurofibromatosis Therapeutic Consortium. It's a consortium of 4 different labs who have animal models. The only scope is to try to test different hypotheses to push forward towards the clinic. So what they did, and this was done about 4 or 5 years ago, they use a prototype, which is not AZD6244, it's a prototype molecule, which worked on the same target and showing that there was efficacy. So then years later, about 2 years ago, when we started the clinical trial with AZD6244, they of course picked that new molecule that was ready and optimized, not the prototype anymore, but the evidence of the prototype efficacy, the proof of concept, was already done. So they picked the right molecule, and now they're showing success because from a recent announcement, this molecule seems to respond very well in patients with plexiforms, and now we see efficacy in about 60% of patients which have more than 20% reduction in their plexiform. So this is really showing success, and is one of the molecules that it can probably make it to the market and can be approved as the first molecule for NF ever. What we stand today is in October they're going to start recruiting for a registration trial, which is the trial that the FDA is requiring, with the right number of patients, with the right mutations, with the right statistics, with everything made perfect to get directly to registration. If everything in this trial goes right, as we expect, because we already have results from a previous trial, if everything goes right, probably we're sitting here next year at the same time saying that, you know, we are in the registration phase, which is the very last stage of a drug before it goes into market and approved for a disease. That will be unprecedented, because that will create a lot of feedback into the industry. They will be more interested in developing NF therapies, because they will see the value, and they will show numbers of what is the market size for NF, what are, you know, those patients taking this drug, and it will create also let's say a building block. It will create already a stage where you can make better treatments. Before, we didn't have any comparison, any gold standard for NF. Now this is creating the gold standard, and if you have a gold standard, it's much easier to compare your own research to. So you know that you need to come up with something which is better than this drug. And sometimes this is the difference between starting a new project and not starting a new project, because sometimes you don't know what are you developing. If you have a chance to succeed, what arguments you need to bring to the FDA, and that will set the stage that the next big win is going to be a molecule that works better than AZD6244, and that will be much easier task to achieve. Does the one drug fix NF entirely? Well, short answer, no, because NF is a very complex disease. It's very simple because it's a monogenetic disease. So there is a mutation that is either in the NF1, NF2, or schwannomatosis gene which cause the disease. But we don't know, apart from that first mutation, what is the second mutation which triggers different tumor types and different phenotypes, different forms of NF. So there's been about 1,500 to 2,000 different mutations detected in NF patients. So you can tell by that number that one size fits all solution, that will not ever happen. So we're gonna, AZD6244 is gonna be a solution for a relatively small patient population, and then from there we need to move to the next and enlarge that to different population size and different type of patients which have different mutations maybe, and create something new for NF1 but also for NF2 and schwannomatosis. In fact, I connect to the next question, which is what is being done in NF2 and schwannomatosis. Well, NF2, we are really making a lot of progress. Before, we analyzed the field, and there were no real good animal model. So Synodus for NF2, for instance, is one consortium who is trying to bring a compound to the clinic, but also another aim, another major achievement of the project will be to create new tools for NF2, for future research. Because those tools we analyzed, we saw that they were not optimal. So that's one of the scope of the project. For schwannomatosis, we are a little bit behind. So we understand what are some of the genes involved in schwannomatosis. We don't know the cause. We don't know why there is so much pain in schwannomatosis patients, and how this is really connects to the two genes. So we believe there will be more basic science. More investment in basic science. One of the projects we had in mind is to gather all the specimen, all the tumors that we have from schwannomas, and start to do a large screening trying to understand what are the targets involved there. Now today we can use next generation sequencing, like analyzing all genomes and see what are all the different mutations that these patients have. But we don't want to go in a fishing expedition trying to see, analyze everything, and trying to capture whatever we picked up with science. We want to go there with a precise question. We need to know what the questions are. We are waiting for clinician researchers to give us the right question to ask to that data, and then we are ready to start. And we are trying to organize also those tumors which are really rare and very difficult to get together different institutions. But I mean, if there is, I believe if there is any entity who can do that, is CTF. So, and we have experience now bringing people together. We're becoming better and better in creating those collaborations. So we believe that we can really impact also in schwannomatosis. What can Cupid do to best help? Well, very easy answer. Raise as much as you can, please. We really need your help. Those projects are very expensive. Think about a research collaborative project for Synodus in F2 is $3 million in 3 years. And that means that even though if we start the project, we need to reserve that money for 3 years. That means that 2015, 16, 17, we need to raise whatever is needed to complete the 3 million. So we're going to start something new for an F1 as well, and those are another 3 projects, probably for $2 million each. We don't know yet the budget, but it's going to be in that ballpark. It's going to be another 6 million that we're going to spend in the next 6 years, and probably there will be more projects activating in next year or this year. So it's really, it's a spinning wheel. Once you start a project, you cannot stop. You really need to raise money. We need to raise money together, and we need your help. Please, please tell everyone about Cupids and Run, CTF. What are we trying to do? You've been amazing, and I've been following Cupids for quite some time, since its inception. I wasn't here when it was the very first year, but I was looking at videos, statistics, and this is so powerful, so great. Keep it up, and really thank you all for all your hard work. I know it's a lot involved. It's only one day, but the revenue and the joy of completing such events in a very powerful way is really has no price. So thank you so much for your hard work. We are on the same side, where we are different sides of the same coin, and the coin really serves to play in the jukebox where the patients choose the song and the patients drive research, and we are there. We are the organization who facilitate research, and you are the one who facilitate research with money, which otherwise we cannot do anything. We stay with ideas on a piece of paper and nothing happens. You make it happen in the lab with the researcher, with really pipetting and creating ideas in the lab. So thank you again, and see you soon next year. hopefully with very great news about this new trial and probably NF2 and news in others new project at CTF. Thank you.